Today we're going to discuss the important concept of meiosis. I'm not going to review mitosis, but you have a lab on that, so that should allow you to review that. If not, review your notes from introductory biology. Here's the cell cycle. The M phase is for cell division or mitosis, and similarly, meiosis works in a similar way. We're mainly interested in that. The pink pie slice there. The S phase is DNA synthesis or DNA replication. That's also important to us in genetics. Here are features of the cell cycle. I'm not going to read it, but you can read it over yourselves. And this shows what happens with numbers of chromosomes, their number of chromatids, number of DNA molecules in dividing cells. Homologs are the chromosomes that have the same numbers. They are identical in chromosome morphology, such as our 2 chromosome 1s, our 2 chromosome 2s, etc. You get one from each parent. They're the same size, and importantly, they have the same genetic information. So at the top is an unduplicated chromosome. Below that is a duplicated chromosome as the type of chromosome that would enter into cell division. And at the bottom is an actual photograph of a duplicated chromosome. The 2 sister chromatids are identical copies of one another. There's a picture of a metaphase chromosome. We mostly see chromosomes during metaphase of the cell cycle because they are most constricted then and best to view. This is a karyotype on the left in the black box there, showing the chromosomes lined up in homologous pairs, and this is from a female, as we can see by the 2 X's in the lower right-hand corner. The number of chromosomes, remember, referred to as autosomes, and the others as sex chromosomes. The anatomy of a chromosome, so to speak, the main constriction is the centromere that contains the kinetochore where the spindle microtubules will attach. The ends of the chromatids are capped with telomeres, which are DNA sequences and associated proteins indicating to the cell that that is the end of the chromosome, and the chromatids then contain the genes, the DNA with the genes, that is. Chromosome morphology, there are 4 types of chromosomes based on the position of the centromere along the chromatid. So if the centromere is right in the middle of the chromatid, it's referred to as a metacentric chromosome, like the second from the top. If it's slightly off-center, yielding a distinctive short and long arm of the chromatid, it's submetacentric. If it's close to the end, but not quite at the end of the chromatid, it's called acrocentric. And if it's at the end of the chromatid, yielding a one-armed chromosome, it's called a telocentric chromosome. So, overviewing meiosis, process has 2 cell divisions, and 1 diploid cell can yield up to 4 haploid cells. Meiosis 1 is a reductional division, meaning the number of chromosomes is cut in half. So after meiosis 1, the cells are haploid. Meiosis 2 then, the events of meiosis of this stage are about the same as mitosis, except it occurs in haploid cells. And prophase 1 takes up to 90% of the time for meiosis. That is because crossing over recombination occurs during prophase 1. So a visual representation of what happens in a cell that's 2n equals 4. After meiosis 1, we have 2 haploid cells, but the chromosomes are still duplicated. Notice they're like X-shaped and not bar-shaped. And then meiosis 2 separates the sister chromatids in those haploid cells. And then we end up with haploid cells with unduplicated chromosomes at the end of meiosis 2. Prophase 1 is long and complicated because of crossing over or recombination. And this ensures genetic variation. So each autosome that we pass on is a unique combination of paternal and maternal alleles. And for females, that's the same with the X chromosome as well. For males, of course, the X and the Y do not undergo crossing over. They're too different to do so. So prophase 1 has been broken down into 5 stages known as leptotene, zygotene, pachytene, diplotene, and diakinesis. Sometimes the suffix "-nema" is used instead of teen. So I've seen leptonema, zygonema, etc. It means the same thing. And here are pictures of cells and what's going on in the nuclei during middle and late prophase 1. And again, late prophase 1. So let's start talking about leptoteen. The chromosomes begin condensation, and the rest of the cellular apparatus behaves as it does in mitosis. Here the chromosomes are still long and thin, however, and they're held to the nuclear envelope by their ends via structures called attachment plaques. And the attachment plaques move the homologs toward one another. And this is sort of one of my crude drawings of how this works, where the circle is the nuclear envelope, the dots, black dots, are the attachment plaques, and notice how it's moving this pair of homologs toward one another. What it looks like in a real cell nucleus. The chromatin's beginning to condense and kind of has a spaghetti-like appearance. The next phase of prophase 1 is zygotene, where synapsis occurs. This is the pairing of the homologous chromosomes. So bivalent is the term used for a synapse pair of homologs. A proteinaceous structure called a synaptonemal complex holds them together. And it's composed of 2 lateral, 1 central, and numerous transverse elements. As we can see here, the blue structure running down the middle is the synaptonemal complex. The recombination nodule is where breakage will occur. That's where a chiasma is. And then the red fibrous stuff is the DNA from the 2 homologous chromosomes. So now the 2 homologs are together and held together by the synaptonemal complex. Attachment plaques are still present, and they're starting to look a little more condensed now. Pachytene, here the chromosomes shorten and thicken even more and become more visible. Since the sister chromatids are visible, and there's 4 of them per bivalent, the bivalents are sometimes referred to as tetrads. And crossing over takes place here. The chiasmata are points of exchange where the homologs break. Chiasma is singular for that word. And when the synaptonemal complex dissolves and the homologs begin to repel one another, that's the end of pachytene. Here we can see 4 sister chromatids and 1 chiasma. There's a real cell nucleus showing it. Onto diplotene. Here the bivalents begin to shorten and thicken. The attachment plaques are lost as the nuclear envelope dissolves. The chiasmata become quite visible here. So we can see there's 1 chiasma. That bivalent's got 2 of them, places where the 2 homologs touch one another. And here we can see, especially at the bottom, 3 chiasmata in this 1 tetrad. Diplotene. You can see here's a cell nucleus with several bivalents in it. Dikinesis, here the chiasmata terminalize or move to the ends of the chromatids. Bivalents look like a ring or a figure 8 if there's 3 chiasmata. Spindle fiber attachment occurs at this point, but now the nuclear envelope's gone. And here we see our nuclear envelope disappearing with the chiasmata in the ends or the middle and ends of these bivalents. And here are bivalents in dikinesis. And so this reviews prophase 1, where they're saying that crossing over starts with synapsis in zygotene and ends with the repulsion of the homologs in diplotene. Some more pictures of meiosis 1 and 2. You go on to metaphase 1 of meiosis. Here, bivalents line up along the equator of the metaphase plate. The spindle apparatus completes formation. Here's the spindle apparatus here. The light things running down the middle are the chromosomes, and the darker things attached to them are the spindle fibers, which of course are protein fibers. And then the centrioles or centrosomes are the places where the opposite end of the microtubules are anchored and will become the nuclei of the daughter cells. So here are the chromosomes lined up in the middle of the metaphase cell. Anaphase 1, the homologs are separated. Independent assortment occurs, which refers to which homolog going to which daughter cell is random. Independent assortment ensures 2 to the n possible gamete combinations, where n is the haploid number. So for our species, that's 2 to the 23rd, or over 8 million combinations of unique genetic combinations of gametes. And this shows for a cell that's 2n equals 6, how independent assortment can function to produce many, many different combinations, unique combinations of chromosomes within the gametes. And here we see anaphase 1 with the homologs being separated from each other. In telophase 1 then, chromosomes move to opposite poles. Cytokinesis occurs simultaneously. 2 haploid daughter cells result, but the chromosomes are still duplicated, so we need a second cell division. And here are the 2 daughter cell nuclei, metaphase and anaphase 1. Review of some of what's going on in the various phases. Interkinesis is a brief stage in between meiosis 1 and 2 with no DNA replication. So now we've got haploid cells with duplicated chromosomes that enter into meiosis 2. Prophase 2 is similar to mitosis, and the chromosomes condense and the nuclear envelope dissolves and the spindle apparatus forms. In metaphase 2, it's similar to mitosis. Same with anaphase 2, where the sister chromatids are separated and not the homologs as in meiosis or anaphase 1. Telophase 2, again, the same stuff occurs, cytokinesis, as the chromosomes reach the daughter cell poles. So here's prophase 2. You can see the condensation of the chromatin into chromosomes has begun. Metaphase 2, the chromosomes line up on the cell plates of both these haploid cells. Some diagrams of what's going on. Anaphase 2, the sister chromatids are separating from one another. Second mitosis. That's shown here in the right panel. And then telophase 2, we've now got 4 haploid daughter cells. And this is a table from your text outlining what happens. I'm not going to read it, but if you can recall this on an exam, you'll do fine for meiosis. Let's compare the anaphases. So they're quite different between meiosis 1 and meiosis 2. Anaphase 1, homologs are separated, so an independent assortment occurs, and after this, the cells are haploid. Anaphase 2, sister chromatids are separated, just like in mitosis, except that occurs in haploid cells in meiosis 2. Now, proteins called cohesion proteins hold homologs and sister chromatids together, and a enzyme called separase breaks these down during the anaphases to separate either the sister chromatids, such as at the top there in mitosis, and really the same thing happens in meiosis 2, and then at the bottom in anaphase 1 and anaphase 2 of meiosis. And this compares both cell division in meiosis with mitosis and the end results. I mentioned genetic variation. It's the raw material for evolution by natural selection. Populations with low levels of genetic variation are at risk of extinction, and most populations that have been assayed have high levels of genetic variation. And during meiosis, genetic variation is maintained by 2 events, that is crossing over during prophase 1 and independent assortment during anaphase 1. So reviewing crossing over for a moment. So a germ cell icon A there goes through DNA synthesis or DNA replication, and now our chromosomes are duplicated and ready to enter into cell division. Notice each chromosome has 2 chromatids now. The sister chromatids are copies of each other. But before cell division occurs in meiosis 1, of course, we have crossing over, and now we've got some unique combinations of gametes, such as big A, little b, and little b, big A chromosomes, chromatids at this point. At the end of meiosis, then we've got 4 genetically different cells here. And the top and the bottom ones are called parental or non-recombinants because they have the same genotype as the parental cells that entered into meiosis, and the ones in the middle that are bicolored are recombinant or non-parental gametes or cells. This shows that whole thing start to finish. Crossing over ensures genetic variation, as we see here, by creating new combinations of paternal and maternal alleles. Now, in our species, of course, in males, spermatogenesis is the formation of sperm that includes meiosis, but it's not limited to meiosis. It begins at puberty for boys and continues until we die. In women, gametogenesis is called oogenesis, and interestingly, it begins before birth, where about 2 million primary oocytes are held in meiosis 1. Then meiosis 1 is completed just prior to ovulation. By then, about 30,000 primary oocytes remain. And then meiosis 2 isn't completed until fertilization of the secondary oocyte. And so by then, only 400 to 500 ova are ovulated by women throughout their life span. So this is a comparison of the two. So in males, spermatogenesis, from one spermatogonial cell, we get 4 haploid sperm. For women, for one oogonial cell, we get 1 haploid ovum. And the other 2 are polar bodies, which are degraded by the cells. It's just the extra DNA. So in males, again, primary spermatocytes enter into meiosis 1, and this occurs constantly once we reach puberty. And the results of meiosis 1 are secondary spermatocytes, which are now haploid. They undergo meiosis 2 to produce spermatids, which then mature into sperm. And the sperm are only a haploid set of DNA, nuclear DNA, excuse me, and no cytoplasmic organelles or anything like that. In women, meiosis primary spermatocytes enter into meiosis 1 prior to birth, and then meiosis 1 is delayed or stopped during birth and childhood. And then when you start ovulating, these secondary oocytes are what is ovulated, and the polar bodies are destroyed by the cell. And then should that secondary oocyte become fertilized, it completes meiosis 2. Now, older women have problems with having gametes with missing or extra chromosomes, and that's because, remember, the spindle apparatus forms before you are born, so it is just as old as you are, if not a few weeks or months older maybe, and these spindle fibers can become brittle and break, and therefore you can have eggs with extra or missing chromosomes. So when a woman of 35 years old or older is pregnant, doctors want to do an amniocentesis where they karyotype the fetus to see if there are any chromosomal abnormalities due to this. And that's the end.